Good morning students. In this video we are going to discuss about some important subchapters from the main chapter, 8th chapter, Cell the Unit of Life. The subtopic or the subchapter is prokaryotic cells. Make sure one point must be clear to everybody, which cells or organisms are called as prokaryotic cells. Organisms are cells which do not possess well-defined membrane-bound cell organelles in them. They are called as prokaryotic cells. Understand? Bacteria, blue-green algae, mycoplasma and PPLO. These are all examples for prokaryotic cells. In these, the very important one is about bacteria. We are going to discuss in this subchapter in detail. And one organism is newly mentioned here. It is PPLO. What is the full form of PPLO? Pleuropneumonia-like organisms. Might be you are seeing the structure of PPLO on the screen, right? Then generally these prokaryotic cells are having some specific characteristics. What are those? These are rapidly multiplying than the eukaryotic cells, and they are very small in structure, and they may be even greatly vary in shape and size. There are 4 different types of bacteria are present in the nature, which we have already discussed in previous chapters. Once again, let's have a discussion about those. Some bacteria are in rod shape. They are called as bacillus bacteria. Some bacteria are in spherical shaped. They are called as coccus bacteria. Some bacteria are in comma shape. They are called as vibrio bacteria. Some bacteria are in spiral shaped or ribbon shaped. They are called as spirillum. Understand? These are the different shapes of bacteria which we can see generally in our surroundings. The organization of the prokaryotic cell is fundamentally similar when even tough prokaryotic cells exhibit a wide variety of shapes and functions. So here on the screen you can able to see a typical eukaryotic cell which is between 10 to 20 μm, and other cells are there. Bacteria generally 1 to 2 μm in size. PPLO is about 0.1 μm in size, and viruses are 0.02 to 0.2 μm in size, right? Then I would like to tell you one important point. All the prokaryotic cells have a cell wall surrounding the cell membrane except in mycoplasma. Now I am going to show you a detailed structure regarding bacteria. See in this diagram bacteria and its parts. So here now I would like to tell you one important point. The fluid matrix is filling the cell in the cytoplasm. So in the cytoplasm there are only non-membrane bound one cell organelle is present, that is nothing but ribosomes, as we learned in previous class. So the fluid matrix is filling the cell. It is nothing but cytoplasm. In the cytoplasm only a non-membrane bound cell organelle is present, that is ribosomes. There are no well defined nucleus in this prokaryotic cell, right? So the genetic material is basically naked. We can able to see with our eyes, and not enveloped by any nuclear membrane. First of all, nucleus is only absent, then how can we see nuclear membrane, right? So generally in eukaryotic cells, nucleus is covered by nuclear envelope, and within the nucleus there is a fluid matrix called as nucleoplasm. In that nucleoplasm genetic material DNA is present. But in the case of prokaryotic cells it is not like that, quite opposite. A fluid matrix is present in the cell called as cytoplasm. In the cytoplasm freely genetic material is present without covering by any membrane. That's why genetic material is naked to our eyes. We can able to see very clearly under the microscope. Understand? So here one more important point. In addition to the genomic DNA, the single chromosome or circular DNA. Many bacteria have small circular DNA outside the genomic DNA. The smaller DNA are called as plasmids. Understand? This is very important. You can see on the screen two types of genetic material. One is plasmid DNA. It is smaller in size. It is called as smaller DNA. Then one more important one is genomic DNA. The genomic DNA generally single chromosome and circular in shape, hence called a circular DNA. You can see blue color one DNA, circular in shape. Understand? As well as you can also see plasmid also in this diagram on the screen, right? I will show you one more important diagram. Please have a look. So in this bacterium, you can see a single circular DNA which is present centrally, and plasmid also can be seen at the corner. Can you able to see? Plasmid is nothing but one more DNA. It is popularly called as with a single name, that is nothing but smaller circular DNA. What is that? Smaller circular DNA. Since it is circular in shape, called as smaller circular DNA. The smaller circular DNA is called as plasmid. But center of the cell there is a genomic DNA. It is also popularly called as single chromosome circular DNA. Then one important point. One of such bacteria is resistance to antibiotics. Understand? So generally the plasmid DNA converts a certain unique phenotypic characters to such bacteria, and in higher classes you will learn that these plasmid and characteristics and about plasmid DNA is used to monitor bacteria transformation with foreign DNA. Understand? Same concept you are going to learn even in higher chapters, in next continuation chapters. And one more information regarding this one: nuclear membrane is found in eukaryotes, right? So we have discussed this point in above concept. No organelles like ones in eukaryotes are found in prokaryotic cells except the ribosomes. As we discussed earlier, there are no other membrane bound defined structures in the cytoplasm of the bacteria other than ribosomes. Prokaryotes have something unique in the form of inclusions. These inclusion bodies are meant for reserving the different kinds of materials. Make sure this point must be clear. About inclusion bodies, we will learn very detailedly in the end of the chapter, or even in this video also we are going to discuss. A specialized differentiated form of cell membrane called mesosome is the characteristic feature of prokaryotes. These mesosomes are present within the cytoplasm and towards the center of the cell, emerging from the plasma membrane. So these mesosomes are present in the bacterium exclusively. We will learn in this video only in detail in the next continuation chapter. They are essentially enfoldings of the membrane. Which membrane? Cell membrane. It is also called as plasma membrane. Understood? Now, students, I am going to discuss with you all a very important concept regarding cell envelope and its modifications. Students, please watch this bacterium diagram on the screen. It is showing three different colors: blue, dark gray, and light yellow color, right? So what are those three? These three are nothing but different layers which presents on the surface of the bacterium or prokaryotic cell, right? In the previous chapter we had a discussion. Bacterium is a prokaryotic cell. It has a tough cell envelope on the surface. Most of the prokaryotic cells, particularly the bacteria cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bounded three-layered structure. Understand? So what are those? The outermost one is glycocalyx, and followed by cell wall in the middle, and followed by cell membrane towards the cytoplasm. So plasma membrane is the inner membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Understand? Bacteria can even be classified into two groups on the basis of the difference in the cell envelopes and the manner in which they respond to the staining procedure developed by a great scientist. Scientist full name is Hans Christian Graham. So this Hans Christian Graham discovered one different type of procedure to observe the clear structure of bacteria under the microscope. The procedure is called as staining procedure. So he have taken up some gram strain or called as positive bacteria, also called as gram positive bacteria, and bacteria those who do not take up or respond to stains, those are called as gram negative bacteria. Understand? So we have taken some chemicals like crystal violet to stain this process, and after staining with a sample of water with the crystal violet, and bacteria are responding to this stain, and those bacteria are called as gram positive bacteria, and some bacteria do not take up this stain and do not respond and not visible under the microscope. Such bacteria are called as gram negative bacteria. So in such a way, Christian Graham discovered this procedure. Which procedure? Staining procedure for bacteria. Understand? Next, one more important point I would like to tell you here. See in this diagram on the screen very clearly how the DNA or genetic materials differ from one to another. Circular DNA can be seen very largely in the center. Next, plasmid DNA is present aside. Understand? So at the same time, you can also able to see glycocalyx in this diagram very clearly. Let's discuss about glycocalyx. Glycocalyx differs in composition and thickness among the different bacteria. Understand? So it is not unique in all. It is a unique characteristic, can't be seen in all the bacteria in the same nature. It is vary. The thickness is vary from one bacterium to another bacterium. Understand? In some bacteria it is thick, and in some bacteria it is thin. Understand? Which one it is? Glycocalyx. It could be a loose sheath and called a slime layer. Understand? In some other bacteria it can be as a thick and tough layer. It can be called as a capsule. Make sure these 2 points must be understand and remember forever. I repeat once again, glycocalyx in some bacteria it is as a loose sheath called a slime layer, whereas in some other bacteria it is thick and tough called as capsule. Understand? Next, let us talk about the middle layer. Just below the glycocalyx, one more important layer is present, that is nothing but cell wall, which is in the middle between glycocalyx to plasma membrane. So the cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. Understand? So these are all functions of the cell wall. So cell wall is mainly performing some important function like determines the shape of the bacteria. What are the different shapes of the bacteria? Like spiral shaped or ribbon shaped. Next, comma shaped. Next, spherical or round shape. Next, comma shape. All these shapes are depend upon presence of cell wall only. So the cell wall is determining the shape of the bacterium, and it is also providing strong structural support to prevent bacterium from bursting or collapsing. Understand? So next, let us talk about plasma membrane, inner membrane. You can see on the screen plasma membrane. or it is also called as cell membrane, which is present just below the cell wall. Understand? So here the plasma membrane is semi-permeable in nature. What is meant by semi-permeable nature? Semi-permeable nature, it is also called as selectively permeable nature. So the membrane allows only the certain minerals and molecules from outside to inside and inside to outside, vice versa. Understand? Because it is having the capacity to filter the unwanted substances without entering into the cell. Understand? And it is interacting with the outer side world also. So which membrane is interacting with the outside world? It is nothing but plasma membrane. Understand? Because it helps in transportation. Please remember this point. Please make a note also. It is very important. So plasma membrane helps in transportation. Understand? This membrane is similar in structurally to the plasma membrane of eukaryotic cell. So the plasma membrane structure is same both in prokaryotic cells and as well as eukaryotic cells also. So that plasma membrane structure in detail we are going to discuss in next video. I will make one separate video regarding plasma membrane. Understand? Don't worry about it. A special membrane structure is emerging or forming from the plasma membrane. Those are called as mesosomes. Understand? These mesosomes are forming from the plasma membrane. They are extensions of the plasma membrane extending into the cell. To be frank, in towards the cytoplasm. Understand? These extensions are in the form of like vesicles, tubules and lamellae. Understand? These plasma membranes extensions are mesosomes. These mesosomes are not in the same shape in all the bacteria. They are in different shapes. Understand? Some bacteria may consist mesosomes in the form of vesicles. Some bacteria may consist mesosomes in the form of tubules. Some may consist in the form of lamellae. Understand? They helps in different functions. What are the different functions? Let us see. Here in this you can see mesosomes. Mesosomes are forming from the plasma membrane. So the yellow color one is plasma membrane. So from the plasma membrane mesosome is forming. Understand? The green color one is cell wall. So outside the cell wall one more layer is present, that is nothing but glycocalyx. Understand? So now I will show you one more diagram on the screen. Please have a look. So this is nothing but mesosome which is forming from the plasma membrane. Understand? The reddish one is cell wall. Below the cell wall there is a membrane, plasma membrane. Plasma membrane is extending into the cell, towards the cell. Those extensions are called as mesosomes. You can see mesosome structures very clearly in this diagram. Understand? Those are just like tubules, vesicles and lamellae. You can see tubules here. They are just like in the shape of tubules. Understand? So let us see functions of the mesosomes. Cell wall formation. Mesosomes generally helps in cell wall formation in the new daughter cells, and as well as DNA replication and distribution of daughter cells also. And they also helps in respiration of bacterium, and secretion process in bacterium also will be takes place by mesosomes only. Okay, what's the role of secretion of different kinds of enzymes? So to increase the surface area of plasma membrane, and enzymatic content is producing by the mesosomes. So with that help, the plasma membrane can be extend on the surface. Understand? In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm are called as chromatophores. These chromatophores are generally consisting pigments, color causing pigments. Understand? Cyanobacteria is popularized as blue green algae. It is also performing photosynthesis process, right? So this photosynthesis process should be done with the help of some color causing pigments, like especially chlorophyll. Chlorophyll is a color causing pigment which is present in cyanobacteria, helps in photosynthesis. Those pigments are present in the a specialized structure called as chromatophores. These chromatophores are exclusively present in the cyanobacteria. Have you understood, ma? Next, one more important, please watch the diagram on the screen. Bacteria generally moving from one place to another place with the help of locomotory organs. Understand? So bacterial cells may be motile or even non-motile also. Make sure this point must be clear. Some bacteria are motile, moving with the help of locomotory organs like flagellum, right? Next, some are non-motile. They sticks to the substratum or even ground or on to the rocks, etc. with the help of one more structure that is nothing but called as fimbriae. Understand? Now let us see, if the bacteria is in motile in nature, they have a thin filamentous extensions from their cell wall called flagella. So flagella is a thin filamentous extensions from which layer? Cell wall. Understand? So in previous diagram I have showed you cell wall, right? So from the cell wall a thin filamentous extensions are forming. Those are flagella, plural, singular flagellum. Understand? Bacteria show a variety of number of arrangement of flagella. So this flagella shows a unique characteristic. Generally, flagellum singular has three main parts. What are those three main parts are present in a single flagellum? In bacterial flagellum. First one, filament. You can see on the screen yellow part. It is filament. It is helical in nature, right? Next, another one, hook region. The blue color part is nothing but hook region. Understand? Next, one more important, basal part. Basal part you can see which is connected to cell wall. The way how basement is very important, the way from basement total uh, building is light, at the same time from the cell wall a portion of the flagellum which is connected or jointed, it is called as basal part or also called as basal body. Understand? So these are the three important main parts of the flagellum in bacteria. So now those you can see very clearly. Next, the filament is long portion and extends from the cell surface to the outside. You can see on the diagram also, flagella are a long projections. In those, especially the filament is a longest portion and extensions from the cell surface into the outside. Understand? Besides flagella, there is a one more important structure, pili. You can see on the screen yellow color projections. Next, one more, it is nothing but fimbriae. Fimbriae also can be seen. The dark gray color structures are called as fimbriae. Understand? So these are also playing a great role in bacterium cell. Let us see their role. They are present on the surface of the bacteria, but they do not play any role in motility or locomotion. Understand? Three different colored projections can be seen on the surface of the bacterium. One is flagellum, just now we had a discussion. Next, pili. Understand? Yellow in color. Next, fimbriae, short ones which are just like a brush bristles. Understand? Dark brown color here, but they are not playing any kind of locomotion of the bacteria. Okay, now let us see what are those functions. The pili are elongated tubular structures made up of a special protein. Understand? Next, the fimbriae are small bristles like structures which are present on the surface of the bacterium. So bristles like fibers are sprouting out of the cell. Understand? In some bacteria, they are known to help attach the bacteria to rocks, in streams, and also in the host tissues. Example, E. coli bacteria in the small intestine of human beings. Understand? So these are the different unique points regarding flagellar locomotion, especially regarding flagellum of bacterium. Next, one more important sub concept I am going to discuss in this video. It is nothing but inclusion bodies and ribosomes. Students, you can able to see inclusion bodies structure on the screen right now. So here students, see this one. All the spherical shaped non-membrane bound structures which are freely present in the cytoplasm, they are called as inclusion bodies. Understand? Now, let us see one more important information regarding inclusion bodies. They are meant for reserving the or preserving the materials in prokaryotic cells. They are also meant for storage. They are present in the cytoplasm of the bacterium. Understand? So reservoir materials in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. Understand? So these are not bounded by any membrane system and freely present in the cytoplasm. Understand? So what are the different examples for inclusion bodies? They are like phosphate granules and cyanophysin granules and glycogen granules. Understand? Then gas vacuoles are found especially in some other bacteria. Those are as follows, like blue green and purple and green photosynthetic bacteria. Understand? So gas vacuoles role is nothing but respiration. They helps in storing of gas. They interacts with the outside world for exchanging of gases. Understand? So these gas vacuoles can be seen exclusively in this following bacteria only. Understand? So now students, you can see some important information regarding inclusion bodies. In this diagram you can able to see clearly about one important structure. Apart from inclusion bodies, ribosomes are also present in the bacterium. So the ribosomes are non-membrane bound structures. They are 70S type of ribosomes. There are 70S type of ribosomes. Understand? So about ribosomes I am going to discuss in detail in this video. In ribos, make sure one point must be clear to you all. Ribosomes are non-membrane bound structures. They are present in the cytoplasm of both eukaryotic cells and prokaryotic cells. Understand? And these ribosomes are not only present in the cytoplasm, they are also present in some other cell organelles like mitochondria and chloroplasts in plant cells and endoplasmic reticulum. Especially on the rough endoplasmic reticulum, these ribosomes are present. Understand? Especially let us talk about prokaryotic cells ribosomes. In prokaryotic cells, the ribosomes having specific characteristics. Their size is about 15 nanometer by 20 nanometer and are made up of two subunits. What are those two subunits? You can see on the diagram on the screen. One is 50S unit, the second one is 30S unit. One is small unit, one is big unit. Understand? Or large unit. The small subunit is 30S and the large subunit 50S, and together forms as a 70S type of ribosomes in the prokaryotic cells. Understand? Ribosomes are meant for synthesizing the proteins since they are called as protein factories. This is very important for NEET examination and KSET examination. Please make a note. Ribosomes are the site for protein synthesis. Understand? Several ribosomes may attach to a single mRNA. M stands for messenger RNA. Understand? Several ribosomes may attach to a single mRNA and form a chain called as polyribosomes or polysomes. This is also very important for competitive examinations like NEET and KCET. I repeat how the polyribosome is forming. Please listen it very carefully. Number of ribosomes which are present in the cytoplasm of the bacteria are attaching one by one and attaching to a after attaching this chain of the ribosomes as a chain of the ribosomes. These ribosomes which are in the form of chain by attaching one by one, these are attached to a single mRNA. Understand? So one ribosome, another ribosome, just like a chain, they are attaching, they are forming. So this ribosomes chain is attaching to single mRNA in the cytoplasm of the bacterium cell, and in such a way polyribosome is forming. The name itself is stating that poly means many. Many ribosomes are attached to form the polyribosomes. It is also called as polysome. Understand? The ribosomes are having a specific characteristics. Ribosomes are generally translating the mRNA into proteins. Understand? The ribosomes of the polysome, it means the ribosomes which are present in the polysome, they helps to translate the mRNA into proteins. Understand? On the screen you can able to see ribosomes, small subunit 30S, large subunit 50S together forms as a single ribosome. In the bacteria it is nothing but 70S type of ribosome. Then one more interesting point I would like to tell you with this context. 80S type of ribosomes are present in the eukaryotic cells. Please make a note, this is very important. As I told you, ribosomes are meant for synthesis of proteins. They are called as protein factories. Understand? The ribosomes which are present in the polysome, they are translating the mRNA into proteins. So in such a way, proteins are forming. Understand? So students, with this I conclude this video. Hope you understood this one. In the next video, we will discuss about in detail regarding eukaryotic cells. Thank you one and all.